At the other end of the spectrum is really thinking about how we can use the cellular machinery for therapeutic purposes. And at the core of some of that is a biochemist at Brigham and Women's Hospital in our Center for Regenerative Medicine, Dr. Yick Fong. Yick.
Okay, so it's going to be a little different, so sit tight. So my lab is really interested in understanding how transcription factors control the stem cell fate and the ability to differentiate into virtually every cell type in the human body. But I guess in a broader sense, what we really want to know is how transcription factors control cellular identity and cell function. So what I hope to achieve today is the approach that we use to address these questions you generally find applicable in addressing any gene regulatory questions you may have in development and in diseases. So, why should we care about how transcription factors work? I'm going to tell you a little bit more later, but in short, they're the main drivers of cellular identity, which in turn dictate cellular function. For instance, what separates a skin cell, a stem cell, and a cardiomyocyte, at the most fundamental sense, is the difference in gene expression patterns. These distinct gene expression patterns are in turn controlled by usually a handful of cell type specific transcription factors, which sometimes we call master transcription factors. It's pretty remarkable to think about it. And just to further illustrate how important and powerful these transcription factors are, these days, you can pretty much interconvert between these three obviously very different cell fates. For instance, turning a skin cell into a stem cell by expressing the now very famous Yamanaka factors, or directly convert a skin fibroblast to a cardiomyocyte by simply expressing three cardiogenic transcription factors, namely GATA4, MEF2C, and not shown here, TBX5. But one thing I want to point out is that this conversion or reprogramming process from a skin cell to heart muscle cells right now is exceedingly slow and inefficient. And the bigger problem is that it's very hard to control. This is largely due to the fact that we really don't have a good idea of what these transcription factors are doing during this reprogramming process. So as a result, as of today, the process is extremely inefficient and stochastic, and that's just a better way of saying that it's still very random. So I think, what is it? So there's obviously a need to better understand how these transcription factors work. So let me see. So let me back to the things. So I think right now what we have is we have these induced cardiomyocytes, but what we really don't know is how faithful they are in recapitulating the normal tissue functions. So as a result, there's this ongoing debate about whether these cells are even meaningful for using in drug screening or chemical library screening. And I think the next level is how effective they are or safe for transplantation therapy. I mean, clearly there are some successes, but there's obviously room for improvement. And I argue that in order to close this gap, we really need to better understand the molecular mechanism by which these transcription factors control gene expression, which in turn control cell fate, that in turn control cell function. So, my job here today is actually present a case for a better need to understand how transcription factors control cellular identity and function. These days, typically, when people talk about wanting to study transcription factors, they usually talk about doing a lot of ChIP-seq and RNA-seq. What these high-throughput analyses give you, as the word high-throughput suggests, is that it gives you tons of long, exhaustive list of genes that may or may not have anything to do with your transcription factors. So in short, they don't address mechanism. So what I argue is, again, that there's a better need to understand how transcription factors control these processes. So what is the method that we use? It's pretty straightforward, I think. We use biochemistry to recreate, or in a more technical term, reconstitute the very complex process of gene expression in vitro in a test tube. So I'm not going to go into too much details about what's going on here, except to say that it takes more than 100 different protein components all coming together in a coordinated fashion on a piece of DNA before transcription can happen. And so, of course, the key question here is, and has always been, is that how genetic information encoded in our genome is accurately interpreted by transcription factors. And clearly, as you see in this slide, transcription factors clearly play a central role. It recognizes specific DNA sequences on the DNA promoter, which we call enhancers, but they can't do it all by themselves. They need to recruit a whole host of factors, for example, chromosome remodelers, histomodifiers, to open up the chromatin to facilitate gene expression. But they also recruit another class of factors called co-regulators, which essentially function as molecular adapters that help transmit instructions from transcription factors all the way to the transcription apparatus, which then goes on to make the messenger RNA. So these black arrows here really represent only a tiny fraction of all the protein-protein and protein-DNA transactions that have to happen before transcription can initiate. So think of these arrows as very important checkpoints for gene regulation. What these checkpoints do is it gives cells the ability to turn on the right sets of genes in the right place, at the right time, and at the right level. In fact, more often than not, it is the loss of this precision in gene regulation that causes many of the human diseases and disorders. So what my lab has is the expertise to express all these 100 or so different components separately, and then by putting them back together and ask how they click to activate transcription, we now, once and for all, has a very unambiguous, direct way of addressing the exact role of each of these components in gene regulation. So I'm just going to highlight two aspects that I think would be of interest to this audience. First, which is kind of obvious, is that you can use it to study any transcription factors that you like or gene promoters that you want. But I think one thing I want to highlight is you're directly measuring transcription, not a reporter activity which is subject to indirect secondary effects, you name it. The second aspect, I think the most pertinent to this forum, I think is, in a way, I think this assay is built to address disease mechanism. So let me just explain. For instance, there are quite a few mutations identified in GATA4, one of the transcription factors that can turn skin cells into heart muscle cells, that's been linked to congenital heart disease. As far as I can tell, I don't think people know what these mutations do, except the fact that they don't seem to disrupt the binding of GATA4 to DNA. So whatever these mutations are disrupting is disrupting steps subsequent to DNA binding. So you can do all the RNA-seq chips that you want. You're not going to be able to address what exactly these mutations are disrupting. So our assay is perfect for this. Remember, we can express most of these protein components recombinantly. So what you can do is swap out the wild-type GATA4, replace it with the mutant version, and ask, does this mutation, first of all, change transcriptional output? If it does, then can we then identify which of these many different checkpoints are being compromised or disrupted by this mutation? And if we can do that, then now we have a platform, essentially, for drug screening or small molecule, see if we can restore, stabilize this interaction that's not being disrupted by this mutation, or bypass it altogether. So what I hope I've done today is present to you a range of possibilities with which you can use this assay to address really fundamental mechanistic questions in gene regulation in normal cells and the disease state. And with that, I want to thank my funding sources, or as I call it, my lifelines. It's more true than ever these days. I want to thank Charles H. Hood Foundation, the Harvard Simpson Institute for their support, and last but not least, very generous funding by National Heart, Lung, and Blood Institute. And thank you for your attention.